Primary and secondary structure of monocot and dicot stem. Primary structure of monocot stem. The outline of the maze in transverse section is more or less circular. The transverse section of the monocot stem shows the following plan of arrangement of tissues from the periphery to the center. Epidermis. It is the outermost layer of the stem. It is made up of single layer of tightly packed parenchymatous cells. The outer walls are covered with thick cuticle. The continuity of this layer may be broken here and there by the presence of free epidermal outgrowths. Hypodermis. A few layers of sclerenchymatous cells lying below the epidermis constitute the hypodermis. The layer gives mechanical strength to the plant. It is interrupted here and there by chlorenchyma cells. Ground tissues. There is no distinction into cortex, endodermis, pericycle and pith. The entire mass of parenchyma cells lying inner to the hypodermis forms the ground tissue. The cell wall of cellulose. The cells contain reserve food material like starch. The cells of the ground tissues next to the hypodermis are smaller in size, polygonal in shape and compactly arranged. Towards the center, the cells are loosely arranged, rounded in shape and bigger in size. The vascular bundles lie embedded in this tissue. The ground tissue stores food and performs gaseous exchanges. Vascular bundles. Vascular bundles are scattered in the parenchymatous ground tissue. Each vascular bundle is surrounded by a sheath of sclerenchymatous fibers called bundle sheath. The vascular bundles are conjoined, collateral, endork and closed. Vascular bundles are numerous, small and closely arranged in the peripheral portion. Towards the center, the bundles are compactly large in size and loosely arranged. Vascular bundles are skull or oval shaped. Phloem. The phloem is in the monocot stem consists of sieve tubes and companion cells. Phloem parenchyma and phloem fibers are absent. It can be distinguished into an outer crushed protophloem and the inner metaphloem. Xylem. Xylem vessels are arranged in the form of Y. The two metaxylem vessels are located at the upper two arms and one or two protoxylem vessels at the base. In the mature bundle, the lowest protoxylem disintegrates and forms a cavity known as protoxylem lacuna. Primary and secondary structure of dicot stem. Dicot stem have a well-defined layer of epidermis and internal structure such as pith, pericycle, hypodermis, cortex and endodermis. Epidermis. Epidermis is the stem's outermost layer. It is made up of tightly packed elongated parenchymatous cells that show up as rectangle structures in transverse sections. The cells lack chloroplast and are transparent. They have an outer layer of cuticle and inner layer of protective epidermis. The epidermis also has several trichomes or multicellular hairs that cover the cuticle. The epidermis contains tiny pores known as stomata in certain locations that aid in gas exchange. Each stomata has a pair of Goth cells, which are specialized kidney-shaped cells that contain chloroplast. They regulate the closing and opening of the stomata. Hypodermis. 3 to 4 layers of subdermal collenchyma tissue make up the hypodermis. The collenchyma fibers that make up the hypodermis are frequently green in color. Below the stomata, the hypodermis is either non-existent or barely visible. General cortex. General cortex is made up of parenchymatous cells. They close the gaps between cells. The outer cortical cells of the juvenile green-stem have chloroplast and produce food. However, the cortex primary job is to store food. In dicot like sunflowers, there are several longitudinally running oil ducts that go through the cortex, and each oil duct has a channel that is bordered by a tiny glandular cell epithelium. Endodermis. Endodermis it is a one cell thick wave-like layer. The endodermis is located at the innermost edge of the cortex. It is composed of cells with a barrel shape that do not encapsulate intercellular gaps. Pericycle. Pericycle is a thick layered tissue. It is outside the vascular stand and is located inside the endodermis. It is heterogeneous in nature. Parenchyma and sclerenchyma fibers both make up its structure. Sclerenchyma appears as semilunar or semicircular patches called bundle caps. The sclerenchymatous pericycles, also referred to as odd-bast, is the bundle cap that is connected to the vascular bundles phloem, whereas the parenchymatous pericycle is located outside the medullary race. Vascular bundle. Vascular bundle is found as an inner ring of vascular bundles or ustule that surrounds the central pith of the pericycle. The number of vascular bundle is always definite. The phloem is located on the outside of each vascular bundle and the xylem on the inside and a cambium strip is in the middle. Xylem. Xylem can be discovered near the pith or inner part of the vascular bundles. Phloem, it is located on the outside of the vascular bundle close to the pericycle. Cambium. A vascular bundle's cambium is a thin band of primary meristematic cells that lies between the phloem and the xylem. It is called fascicular cambium and intrafascicular cambium. Medullary race. The cortex, pericycle and pith are linked by the medullary race. The cells of the medullary race are more substantial than cortical cells. The ray cells are polygonal in shape. The intercellular gaps are very small. True pith ray cells come into close touch with the phloem and xylem conducting cells. Food and water are transported radially with the aid of medullary rays. Additionally, they move gases from the cortex to the pith and vice versa. Pith. It serves as the stem's center. Polygonal oval or spherical parenchyma cells that encapsulate intercellular gaps make up the pith. The food is stored by the pith cells. In some dicots such as cucurbita, the middle portion of the pith separates to form a cavity.